Thank you for that introduction. So it's Corey Brizzee. It was close. So I'm the director of gene editing at Demeetra, and today I'll be talking about the multiplexing with accuracy, building better CHO platforms with Cas-CLOVER. So a little bit about Demeetra. We're located in Lexington, that's Kentucky, not Massachusetts, where we're known for horses and bourbon. But we're a small biotech company. 80% of us are hands-on research. So we're working with our technologies 24/7, and this is enabling us to optimize these gene editing technologies to put them into our clients' hands. Then we're able to transfer that validated know-how with typical protocols that every scientist would like to use with some ins and outs, so you're very successful once those get into your hands. And this makes the commercial licensing a little bit easier because we have innovative commercial licensing terms as well with these technologies. So a little bit about Demeetra's core technologies. Our first one is our license-free harborin transposase. So this is based off of the piggyback transposase, and it integrates cargo at highly expressed sites, usually in open chromatin and non-coding regions. And we all know that piggyback is very capable of doing large cargo capacities, so up to 200 kb. Our second core technology is our Cas-CLOVER nuclease. So this is a highly efficient dimeric nuclease. So this is using an inactivated Cas molecule fused to our proprietary CLO51 endonuclease. This endonuclease CLO51 is an obligate homodimer. So it actually needs a left and a right monomer to come together to actually make the cut. So CLO51 is a type 2S restriction nuclease, so this actually cuts like one. So it doesn't do the typical blunt cut that Cas9 would produce. This actually produces a staggered 4-base overhang, which is leading to large deletions and high knock-in efficiencies. And we're seeing lower off-target and less translocation events that you would typically see with CRISPR-Cas9. So we can produce these proteins in E. coli, and we can actually do some in vitro cutting assays to look at how this cutting mechanism actually happens and the products that Cas-CLOVER gives as well as Cas9. So we can take an amplicon from genomic DNA. We have our left guide and right guide for Cas-CLOVER. You can see the PAM site here and PAM site here. This region can be very flexible with Cas-CLOVER because this fusion linker is very flexible. So you do have some flexibility in between here. So we can incubate Cas-CLOVER with these two guides and this donor template. And then you have Cas9, and we can just do it with the left guide or the right guide. And you can see the cutting product. So after you actually sequence these, now you can see the 4-base overhang in between the left and the right guide, and the typical blunt cut that happens with Cas9, 3 base pairs from the PAM site. We're seeing improved efficiencies versus Cas9 in CHO cells. So for knocking out, we're choosing the glutamine synthetase gene, where we can incubate with Cas-CLOVER guides. So that's 2 guides. This can be from IDT or Synthago, or a 2-part guide RNA where you actually are complexing the tracer and the crimp RNA together prior to transfection. And we're seeing a very consistent 75% cutting at this loci, and then Cas9 is around 55%. We're also seeing improved knock-in efficiencies. So this was in HEC293 cells where we're knocking in GFP plus a selection marker, and this is around a 7 kb construct. Cas-CLOVER post-transfection, this is prior to selection, we're seeing instantly 10% of your knock-in efficiency, as well where you see Cas9 is only around 3%. So that's around a 3 to 4-fold increase in knock-in efficiency. And then we can post-selection, we can boost that gene editing pool to 70%, and then Cas9 can only get to around 30%. So we want to take Cas-CLOVER and make new commercial cell lines. So we adapted CHO-K1 to suspension, and we're targeting glutamine synthetase because this is a very common system. So with Cas-CLOVER, we can utilize its specificity to actually target the main gene in chromosome 5 and the pseudo gene that's in chromosome 1. And we can do that simultaneously, or we can do that separately if we wanted to. So the blue guides kind of correlate with what will work with the pseudo gene, but the red guides correlate with what will work with chromosome 5, but not the pseudo gene in chromosome 1 based off of mismatches. And this is showcasing how high fidelity and high specific Cas-CLOVER is. So shown here is targeting chromosome 1, the pseudo gene. We're using the blue guides, and we're seeing very good editing efficiencies at that gene, where we're not seeing that with chromosome 5, because these guides are too far apart for those to actually cut that main gene. And then when we take characterization of the indels, we're seeing larger deletions. So these are typically going from 10 base pairs to 28 to 40 base pairs, something that you would typically not see with CRISPR-Cas9. And these cells behave very, very nicely. They have a really good phenotype and glutamine sensitivity. So grown with glutamine, they behave just as normal cells. Without glutamine, these are completely dying within 9 days. So now that we have a good parental cell line, we want to expand and make derivative cell lines for clients, because they may need some other genes knocked out, maybe some other applications for their antibodies of interest. So in this case, we're making a derivative cell line, adding on to the glutamine synthetase knockout and adding a foot-8 knockout. So this would make afucosylated antibodies. So with Cas-CLOVER, we're seeing really good editing at foot-8. So around 80% from a pool 72 hours after transfection. And this is leading to a really streamlined process. When you look at LCA selection, you can do LCA staining and sorting. So prior to sorting, our population is around 98.5% negative in LCA staining. So if you have fucosylation, you would see positive staining. So now that the foot-8's knocked out, you see most of the population is negative. And then after sorting, 100% of that population is negative. So they all have foot-8 knockouts. So then you can single cell clone, and you now have a really nice clonal population and frequency distribution of different clones. So we screened around 48 clones, and this is the indel distribution of these clones. So we're seeing clones that have half exons deleted, full exons deleted. So these are our top clones down here based off of these big deletions. And this indel distribution is quite different than what you would see with CRISPR-Cas9, because you would see most of your population between 0 and 10 base pair deletions. So another thing that's been becoming more of a problem with cell line development is host cell proteins. So host cell proteins can affect the antibody itself in the CHO cell as you're making the antibody, as well as effects downstream to the purification. It might be sticking to your antibody, and that makes the efficacy a little bit lower. So we wanted to target a couple of host cell proteins, doing a couple of different ways. So we targeted 3 different host cell proteins, and we tested 2 guide pairs for each of these host cell proteins. So we can see every pair worked that we designed, and we picked the ones with a purple star. We picked these because they were the highest editing efficiency. So 63%, 44%, and 59%. So now what we did is we multiplexed all of these all together. So we targeted 3 different host cell proteins all at once, and this creates, we can get a lot of clones with a lot of variations in 1 host cell protein's knocked out, 2 host cell proteins are knocked out, 3 host cell proteins are knocked out. So this was our greatest chance to streamline the process and get 3 different, or 8 or 9 different combinations right away. So we single cell sorted these, and then these cells went towards a multiplex PCR, where we were able to identify clones that were just edited in one host cell protein, a couple that were edited in 2, some that were in the other 2, and then some that had potential triple knockouts in 6 to 7 alleles. CHO cells are very alloploidy, so we were seeing that third allele pop up here and there. Then you can characterize these further by taking your clones and looking at relative protein expression. So this is our regular glutamine synthetase knockout control parental line. And then you can see these clones, the protein expression, and you see some that have faint, maybe 20 to 50% knocked out, and then you see some that are fully knocked out. So this helps us validate that these clones aren't producing these host cell proteins. We can also use these CleanCut Cas-CLOVER generated cells to now use it with the license-free harborin transposase to make stable expressing trastuzumab cells. So we're a gene editing company, so we don't have that higher scale Amber system. So we only go to the 24 well deep well plate. So in an unoptimized fed batch with the viable cell density only around 10 million cells per mil, we were able to get our top clones up to 5.5 grams per liter. But the cell-specific productivity is what really stands out. So we're seeing anywhere from 60 to 110 cell-specific productivity, which is very high for harborin that produces 5 to 15 copies in your cells. And these pools themselves were around 3 grams per liter prior to the deep well plates. So overall, Demeetra has a really comprehensive freedom to operate cell line development package. So we have a lot of licensing surrounding the whole entire system. So the CVC has really good inactivated Cas9 IP. We have a nice license with them. We also have other licenses for the CLO51 nuclease, as well as Poseida Therapeutics, which is our sister company. They have this whole entire system for cell and gene therapy. And then Demeetra, as an R&D company, we're making further improvements on this whole entire system, and we have more IP surrounding that. So this is all bundled up in just one license for our clients. So they don't have to worry about royalties or downstream milestones. It's just one simple license. We also have a very good license with ECAC for the CleanCut GS CHO cell line, which is very nice for clients. And the harborin transposase doesn't have a license, so that's even better. So with that, we do have a booth at 708, so you can, if you want to talk more, you can, all 4 of us are here today. Jack, our CEO, is down here. And then we do have a poster the rest of today, A11, with Christian Cummings. Thank you.
Any questions?